What I want to do next is talk about cell membranes some more. Before we talk about transport in detail, I want to talk more about that phospholipid bilayer itself, the structure of it, why it's so important, and it's going to itself allow some things through and not other things, and that's a good place to start with getting things across the membrane. So before we talk in detail about the phospholipid bilayer, I want to remind you of some important terms from chemistry. So hydrophilic and hydrophobic. Hydrophilic, hydro means water, philic means likes, so liking. So hydrophilic molecules are polar. These interact with water. That means they dissolve. That's what's shown over here. Get my laser back here. These are typically ionic compounds and other polar covalent molecules. If you like your chemistry, it can be helpful to know that. These are things because something that's ionic, NaCl, something that's polar covalent bond, H2O, water, glucose, are going to be able to dissolve in water. And you know this from making lemonade or Jell-O, where you stir and stir, and that sugar and the salt are able to dissolve in water because they're polar, they're hydrophilic. They like the water, so they interact with it very closely. That's what these hydration spheres are. That's the water interacting very closely with these molecules based on their chemical structure. On the other hand, hydrophobic means fear of or dislike of water. So lipids are our primary hydrophobic substances. Lipids are made from chains of carbons, typically. These rings are also hydrophobic as well. Here are long chains of carbons. Remember, if you've had chemistry, at least in high school, these represent each a carbon with hydrogens on them as well. Carbon compounds do not dissolve just carbon. There's not polarity here. There's not ionic charge. They do not dissolve in water. So most hydrophobic substances are nonpolar. That means there's no charge to them. Since water is polar, they're not stable together. They stay separate. They don't mix. Oil and water don't mix, right? So here is what that looks like here, these lipid droplets. And you've seen this maybe making salad dressing, oil and vinegar salad dressing, or other examples, oil spills, where that oil stays in separate droplets. It's more stable this way than it is mixed into the water. If you shake up that container, the droplets will get smaller, but still stay separate, and then they'll group back together again. Our lipids are, butters and oils is what you know from your everyday life, with the steroids, and phospholipids are a little bit hydrophobic and a little bit hydrophilic. That's called amphipathic. So phospholipids, they have a nonpolar portion. As you remember, fatty acids as one of the building blocks of phospholipids, and these molecules, phospholipids, are in your chapter, in your book, in lipids, because they are lipids, but they also have a polar portion to them, a hydrophilic portion, a polar head. This is really important for how they function in membranes. This is called amphipathic, meaning both hydrophobic and hydrophilic regions. This is an amphipathic molecule. Proteins can also be amphipathic, and actually transmembrane proteins are, because that's how they can be stable in the membrane. Back to that. So we've got this charged head, and then the uncharged hydrophobic tails, two fatty acid tails. So why does this matter? So when you put these molecules, phospholipids, into water, what are they going to do? Those hydrophobic parts are going to get away from the water. So these molecules spontaneously form one of two structures. Micelles, and these are typically one-tailed molecules. So one-tailed phospholipids, a little different, but you may have heard of micelles. This would be soaps, detergents. Do this. Hydrophobic region is hidden from the water. Water's out here, right? So we're just dumping this into water for some reason. in our bodies, it's because our bodies are mostly water. So that's how this applies to us. The other structure that forms are lipid bilayers. So these bilayers come together with the hydrophobic tails buried in the middle and the hydrophilic exposed to the water. So the part that likes water is next to the water. The part that doesn't like water is hidden from the water. Very convenient and happy. This is spontaneous. No energy is required to do this because this is the most stable, low energy form. And this was probably how the first cell formed. So I should look at this, what this looks like all the way around. I really like this picture because it gives you the visual of what a plasma membrane really looks like from a cross section. These hydrophobic tails are hidden in the membrane, not in the cell. Why can't they be sticking out into the cell? Because our cell is watery, it's aqueous. So those hydrophobic tails don't want to stick down into the water like they are in this situation. So the purpose of this is then to maintain boundaries. We also need to allow things in and out. The phospholipid bilayer itself just refers to this, phospholipid bilayer, literally a bilayer, 2 layers of phospholipids. Then when you have plasma, when you have the proteins and cholesterol in there, that's what's the plasma membrane. So first I want to talk about what can get through just this bilayer, because that's important. Some things can pass through here without a membrane protein to help them, and some things can't. When things can't pass through, they need a protein to help them. So we'll discuss membrane proteins in the next lecture. For just the phospholipid bilayer, a good analogy is like a graded fence that can allow some animals to pass through, but not others. So if a wolf is chasing a rabbit across your yard, the rabbit might be able to slip through this fence, but the wolf can't. So it's like and that size is going to be one important property for getting across this lipid bilayer. So what are some properties of this fence that allow some things to pass through and not others? Again, one thing I already said was size. What allows this to be selectively permeable, meaning some things can come in and some things can't? Well, you should have some thoughts already. Chemical properties are going to matter, right? We've got hydrophobic in the middle here. That is going to be key. Let's go here. I really like this chart. So please spend some time looking at it to make sense of it. This scale, you don't need to know the scale besides, in terms of centimeters per second, is how quickly something can pass through. Low permeability, high permeability. How easily can something pass across the membrane? These are things that can't pass across easily down here. These are things that can pass across very easily. And this is the schematic showing that. So literally showing like how it passes through. These are going to be small things. And this is then telling you what those things are. So let's start with what can pass through easily. Small, we already talked about. And then uncharged, so non-polar. Why can't we have a hydrophilic molecule polar pass through here? It's not going to happen. It can try and it can like start to, but then it's just not spontaneous. It's not going to be energy stable going through this hydrophobic core. There's resistance there, oil and water. So these small things can pass through because they're small and they're hydrophobic. They can just sneak on through there, no problem. So oxygen, carbon dioxide, nitrogen, this is going to be important for respiration. Then we've got these that can pass through to some extent. Small, still, maybe a little bit bigger, uncharged, but some polarity to them. So water is a good example of this. It can pass through some. It's not going to quite as quickly, but it can. We'll get back to water. There is a protein that helps it because we need water to pass across a lot. Glucose can pass through some, not a whole lot though. It's too big. It's also polar, and then it's a lot bigger, right? 6 carbon ring versus H2O is 3 atoms. So we've got bigger things here that are larger, and they're hydrophilic. So that's not going to happen. Then we've got charged ions. These are hydrophilic. These are charged. Na plus. It's not going to be, even though it's small, it's tiny. It can't go through here because the hydrophobic core. It is hydrophilic and it is charged. It's Na plus. So it cannot spontaneously go through that hydrophobic core of the bilayer. The other thing that's not shown on this chart is just large things, right? So a protein, other large items that literally are bigger. So that's why size and scale matters. Phospholipids are pretty small. Something that can't fit through this space, like even come close, is not going to be able to pass through the membrane. That's that the wolf trying to go through a hole this big when the rabbit can. So this allows us to control what's in and out of the cell, just straight up like this. But then we also do need to be able to get other things in. We need to be able to get glucose in for sure. Sometimes we need to be able to get proteins and other things in as well. That's where we need membrane proteins. There's also other types of transport like vesicular transport. So membrane proteins can alter the permeability of the membrane. The phospholipid bilayer itself is selective. The plasma membrane with those proteins in there is also selective, but it changes the selectivity, right? So we'll come back. The next series lecture will be on those transport mechanisms, either not using or using proteins.